Welcome back to my channel Adhyan with Flow. Okay guys, let's begin with a very important topic, linkage and recombination. This is the exam point of view question. Okay, before starting with linkage and recombination, you have to know about what is crossing over. If you are not familiar with this term, then please focus. Guys, here is one chromosome and here is another chromosome, but they are homologous chromosomes. Same genes located at same position. That's why we call it as a homo. Same. Locus means their sites. Guys, during meiosis, what happening? During meiosis, first stage na prophase, metaphase, anaphase, telophase. Remember guys, prophase divided into 5 phases. Remember leptotene, zygote and pectine. You remember this thing? Pectine. What thing is happening here? In pectine stage, they are all crossing over. If you study cell division now, then you can easily know about this thing. So crossing over happening in pectine stage of prophase and meiosis first. Now what is crossing over? Simple. Exchange of genes and it may be genetic material. I am drawing here. This is the one chromosome, there is another. Now here is, they make a bridge form. They make a bridge between two homologous chromosomes. There is formation of bridge. Now with the help of this crossing they exchange their genetic material. Means some part of this chromosome, okay, let me change pen. Some part of this gene goes into this chromosome. Some part of this chromosome goes into this chromosome. Means assume this is the chromosome A and chromosome B. Let's assume this is gene A and gene B. Some portion of gene B goes into the chromosome A and some portion of chromosome A goes into chromosome B. So this is the crossing over. Now with the help of crossing over, how you understand linkage? Now I think you are familiar with chromosome term. Are you familiar with crossing over term? Here is gene A, here is gene B. Here is gene A, here is gene B. Now what happening? We study. Now we study what? Crossing over. Crossing over between same means homologous chromosome. There is gene A and there is gene B. There is gene A and there is gene B. Now what happening in chromosome 1 and chromosome 2? What happening? They exchange their genetic material. They exchange their genetic material. After exchanging of genetic material, what happened with this? What happened with this? I'm changing color, okay, same, okay. Same, okay. In firstly, they contain gene A and gene B, capital A, capital B. But now, after crossing over, they get, look at here. Why? Because capital B goes to this chromosome and small b goes to this chromosome. Now they exchange their what? They exchange their what? They exchange their gene. So, if we segregate this thing, so we found this thing. We found this thing. And this thing is already same. This thing is already same. Wait. Small b, small. Now we get these gamete. Now, how you understand about linkage? Linkage means, guys, when two genes are located closely. If two genes are located closely, then they can't exchange their genes. I'm drawing here. This is the chromosome 1 and this is the chromosome 2. If two genes are attached very closely guys, very closely, if you do crossing over between them there is no exchange of genetic material because they are present very closely. So these chromosomes, these things are linkage. This is the complete linkage. Because two genes are closely located, they can't exchange their genetic material, they can't exchange their genetic things, they can't exchange their genes. But if two genes are located far from each other, far from each other. If crossing over between them na, if crossing over between them, then they can easily exchange their genetic material. So this kind of linkage is what? Incomplete linkage, because their both genes are far from each other. So this is the complete true linkage and this is the incomplete linkage. I think you understand about linkage. Okay, let's discuss about recombination, where I am explaining. Okay. Now what you understand by recombination? This is the one chromosome, this is the another homologous chromosome. Guys, here is gene 1, gene B, small a, small a. Understand? Whatever I am drawing here, you understand? There is 2 chromosome and they carry their genes. What are happening with them? They recombinate with each other, they link, they cross with each other. Now what kind of arrangement now we get? Something like this. Yes or not? Something like this. Are you agree with me? Now what we got? Now segregate these gametes, separate these gametes. We got this is same to same now. This is the parent gamete, this is the same to same. Look at here guys, this is not same to same for their parents, this is also not same to same from their parents, but they are same to same from their parents. Look at here, they are same to same, but these two are not same to same, they are recombinant. I think you are clear with linkage and you are clear with recombinant. Only 50% recombination is there. Why? Because they does not take participation in linkaging and crossing over, nothing. So only 50% gamete will do recombination. Look at here. Here is in your book they elaborate linkage and recombination, guys. They take what? They get 2 chromosomes. I'm not explaining this thing, I'm explaining this thing. They get what? They get, guys, 2, like this manner. Assume na, assume this is the capital Y and capital W, this is the small plus and W plus. What you understand by wild type? This is the pure, guy, this is the parental type. No matter they are writing in small letter, but they are parental. Now they are different, they are not parental. If you cross between them, look at here, let me draw here. Where? Okay, this is the one chromosome and their genes. Guys, you can write down in this manner also and you can write down in this manner also. Okay, now I'm explaining here. This is the one chromosome and this is the another chromosome. Let's cross between them. Now what's happening? If I cross between them, na, they cross with this thing and they cross with this thing. Can I got this thing? One is this and one is this. If I cross between them, one thing I got this and another. Can I draw elaborately? Let's assume this is the capital W, capital Y, capital W, small y, small w, plus plus. If I cross between them, this is a set now, there is a set, they have another set, capital Y, capital W, they are set. If I cross between them, na, they occupy their one offspring, like capital Y and capital W, small y and small w, they goes in one offspring, same na, and rest of, if I cross na, they enter in different offspring. Clear? What happening here? What happening in here? This is the parental or not? This is the parental or not? Yes, they are parental. This is the parental or not? Yes. This is the parental or not? Yes, this is the parental also, and they are also parental. But now look at here, they are not parental. Small y plus, no, this kind of gamete is not here, this kind of gamete is not here, so they are recombinant. So this in your diagram they explain different type and I explain in different manner, but all over they are correct. Okay guys, now guys, in next video we were going to cover about polygenic inheritance. That's all for guys, and we will meet in our next video. Thank you guys, that's all, and don't forget to subscribe my channel Adhyan with Flow and keep learning. Thank you.